Hi, I'm Professor Johnson, and in this video we're going to be talking about Mendelian genetics. So Mendelian genetics are named after this guy over here, Gregor Mendel, who did some of the pivotal experiments on genetics in the late 1800s that led to our understanding of basic genetics. And he worked with these plants known as pea plants, and from this he was able to really come up with two laws that really describe the behavior of genes and the basis of heredity that we now see. So in this video, we're going to be looking at genes and the structure of genes and what genes actually are. And then we're going to go into depth on Mendelian genetics, the experiments that he performed, and the two principles that he came up with, the one being the principle of segregation and the other being the principle of independent assortment. We'll also talk about probability because this is pivotal to our understanding of genetics and really what Mendel was observing were equivalent to probabilities. Now there is a lot more to genetics than Mendel, and we'll get to that in a future video, so I want to emphasize some of the basics here in this video before we get into that. So first of all, looking at what a gene actually is. So genes are located on chromosomes. They're made up of DNA, and they're going to code for specific traits. So this would be, you have this thing within your chromosomes that are coding for eye color, or hair color, or height, or weight, or some sort of behavioral characteristic. All of those things are traits, and all of them can be controlled by one degree or some degree to another by your genes. So now these genes can have different variations of them. So here we can see you have your two homologous pairs of chromosomes, and they have the same gene, the same copy, the same type of gene on them. Here they have two different versions of that gene. So this being the white version of the gene and the purple version of the gene. This is what we call alleles. So alleles are the individual genes within the chromosomes themselves, and so it's the variation of the gene that you actually have. And you can have either your individual that has two different versions of this, or they can have the same version as well. Now genes are actually the DNA, and the traits are properties of the proteins. The proteins that are expressed by the DNA, because the DNA tells your cells how to make proteins, are the proteins then are providing some sort of activity that gives you your trait. They can be an enzyme, they can be a structural protein, what have you, but somehow these proteins lead to your trait that you're actually looking at. In this case, we're looking at the trait for flower color. So we have two individuals that have we have an individual that has two of the same variation of the gene here. So this is a homozygous individual. The heterozygous individual had two different versions. So this was when this was white and this was purple. Or these could both be purple, and that could be a possibility as well. Now we need to talk about dominance and recessiveness. So dominance, when you have an individual that is heterozygous, if one of the traits outshines the other trait such that you only see the one trait and not the other, the trait that you do see is said to be dominant. So in this case, the purple trait was dominant. So when we had one white allele and one purple allele, you ended up with a purple flower. So you had purple being the dominant trait. You can only get the white flower when you have two of the recessive traits. Now, if you have two of the dominant traits, you would also have a purple flower, just like you would in the heterozygous individual. So let's look at how Mendel set up his experiments. So he performed experiments with true breeding white flowers. That meant that when he bred them with other flowers within his group, they only gave birth to white flowers. And he also had a true breeding purple flower group. So every time that he bred these with flowers of the same group, they only gave rise to purple flowers. So he took a true breeding white, a true breeding purple flower, crossed them, and ended up with a purple flower. And this is found in the F1 generation. So this is the filial generation. This is the first generation that he looked at. So filial 1 is what the F1 stands for. And he only got purple flowers with this. He never got a white flower, even though the white flower was one of the parents had the white flowers. Now he went one step further, and he took these plants that only gave rise to purple flowers, and he bred them together. And when he did this, he found that he mostly got purple flowers, but approximately 25% of the time he would actually get a white flower. And this was in the F2 generation that he observed this. So what was going on here? Well, in this parent generation, these individuals were homozygous. They had the same allele for this flower color gene. One was homozygous recessive, that was the white flower, and one was homozygous dominant, that was the purple flower. Now, if we take this and put it into a Punnett square, so these chromosomes here separate during meiosis, and that's what this represents here. So meiosis produces different gametes, and so you have the gametes of one parent up here. So this represents one gamete, this represents the other gamete, and then this would be the gametes of the other parents, and they can produce either this gamete down here or this gamete down here. Now you often see letters being representing the genes that you're looking at. So here the gene for flower color is represented by the letter P, with the dominant allele being the capital letter and the recessive allele being the lowercase letter. Now if we fill in the Punnett square, these zygotes will end up fusing. So each of these boxes represent the gametes that are going to fuse to give you your zygote. So this box here is a zygote, this box here is a possible zygote, this box here is a possible zygote, and this one's a possible zygote. So one possibility would be that the offspring would get one allele from, get this allele from this parent and get this allele from this parent, and so you end up with this being one possibility, this being another possibility, this being your third possible zygote that you'd get, and this would be your final zygote. And when you look at the letters here, you can see that you have a capital P and a lowercase p in all of these, because this parent only had the dominant allele to give, and this parent only had the recessive allele to give. So now let's look at what he did when he crossed the F1 generation with itself. So when he did this, he took plants, and one of the parents' alleles goes down here, and one of the parents' alleles go over here. Now, when we look at these, we can see that the parents no longer have just one type of allele to give. Because they're heterozygous, they can give either the dominant allele, represented by the capital letter, or the recessive allele, and that's same for both of these parents. So the zygote that you get from the fusion of these two gametes ends up having two of the dominant alleles. This one ends up having two of the this one here and this one here has one of each, and then this one has two of the recessive alleles. So this one ends up being capital P, capital P. These end up being a mix of the two, and this one's lowercase, lowercase. So what this box represents here are the possible zygotes that you can get, and therefore the possible offspring that can develop from them in the F2 generation. So we can see purple, 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 white, white, purple, and white, white. So you end up with the three different flowers. So you have a 3-to-1 ratio here of the dominant trait to the recessive, or you have a 1-to-2-to-1 ratio of the homozygous dominant to the heterozygous to the homozygous recessive zygotes. So this really brings us to the principles that Mendel came up with. So his first principle was the law of segregation. And this is just the idea that when you have an individual that has different traits, when he goes to reproduce, the copies of the trait that he has segregate from one another. And what's going on here is you have these genes on your chromosomes, and during meiosis, the homologous pairs of chromosomes are separating. So that's what I'm trying to illustrate here. The homologous pairs separate during meiosis, and now uh, the, um, they're no longer with each other. So it's going to separate, and that produces different gametes, and each of these gametes have an equal opportunity to be fertilized by and bring on the next generation. So the law of segregation is talking about the segregation of the homologous pairs. The principle of independent assortment comes into play when you start looking at how different homologous pairs can separate. So for simplicity's sake, I've only shown an organism with two pairs of homologous chromosomes. So one pair is down here, it's shorter and a little bit darker stained. The other one's longer and lighter stained. You can see that they can line up such that you have this one associating with this one, or this and this one associating with this one. They can also line up such that you have, instead of white associating with yellow, you can also have white associating with green and purple associating with the yellow instead of the purple associating with the green. So from this, you can have four different gametes being produced, one with the white allele on one chromosome and the yellow allele on the other, one with the purple allele on one chromosome and the green on the other, one with the white allele and the green allele, and then one with the purple allele and the yellow allele. Now remember, these are different genes up here, so you're looking at these are the same gene up here, but they're different genes from over here. So this could be a gene for eye color, and this could be a gene for hair color, for example. So let's look at the law of independent assortment in action. So Mendel not only looked at flower color, he actually looked at 7 different traits. And some of them he looked at what happens when you breed an organism that has 2 of these traits at the same time. So one of the 2 of the traits that he looked at were seed shape. So seeds could be either round or wrinkled. And seeds could also be either yellow or green. So he looked at what happens when you have a yellow wrinkled and yellow round, and green wrinkled, green round, and breed them together. So he ended up with a true breeding parent that was just had the dominant allele. So it was round and it was yellow. So this is the dominant trait over here, being round and yellow. He also had one parent that was true breeding recessive. So this was green and wrinkled in the other parent. So he crossed these and ended up with the F1 generation. The F1 generation all showed the dominant traits for both of these. So they were all round and yellow. So now we're going to look at what happens when he self-fertilized the F1 generation and got the F2 generation. So what happens here is that he can have more zygotes than just the 2 that we saw before. So this Punnett square is actually going to be a 4x4 instead of a 2x2. And how we get this is the different zygotes. So this chromosome here is going to separate from this one. So that's the principle of segregation. These 2 are going to separate and they're not going to come back to each other. So this allele separates and this allele separates from this allele. When it does this, it can separate such that the big R and the big Y end up together, and that's what's shown here. So this is one gamete. Notice that these parents have the same genotypes. So this gamete then has the big R and big Y, and it's the same for this one up here. This other way it could segregate is, or the other type of gamete that can be produced when he does this is the little r and the little y, and that's what's represented right here. Or he can have a big R and big Y, or big R and little y, and that's what's represented right here. or he can have a little r and little y, and that's what's represented right here. So now you have 16 different possibilities of what can happen when they fuse together. And that's what happened here. So you can see that this big R contributed to this one, and this big R contributed here, big Y. So you just go down and fill this out. This Y goes into all of these. This Y would go on into all these. You fill all that out, and you end up with the genotypes of the different gametes. So the genotypes are always going to be the letters that tell us what genes, what alleles the organism actually has. The phenotype is how the organism actually looks. So with the what we're looking at here are the genotypes, the letters, what are actually being expressed. The genotypes then are what we see. So you end up with 9 of the gametes, or 9 of the zygotes having the dominant phenotype for both of them. So you have 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 that have dominant round and dominant yellow. You'll have 3 of them that have dominant one of the dominant traits but not the other, and then another 3 that have those flipped around. So here we can see, remember that round was dominant, so we have dominant round, dominant round, dominant round, but recessive because they're all green. Here we have dominant yellow, but the recessive trait of being wrinkled is also being expressed. So you end up with 9 dominant, 3 dominant recessive, 3 recessive dominant, and then 1 out of 16 are going to be recessive. So they're both wrinkled and green. So that's what a dihybrid cross looks like, and how the principle of independent assortment actually applies to it. It's because how these separate isn't going to affect how these segregate. So each of these different 16, or each of these 4 different gametes are possible, and 16 different zygotes are possible then as well. Now this gets us into probability, and when we talk about probability in genetics, there's 2 important things that we need to take into consideration. One is the rule of addition, and the other is the rule of multiplication. So the rule of addition is saying that when you're looking at the probabilities of mutually exclusive events, the probability of both of those happening is the addition of those 2 being added together. So looking just at this Punnett square over here, the probability of getting a heterozygous individual is the same as if you were to add up the probability of all the heterozygous individuals in here. So you have a 1 in 4 chance of getting this probability, a 1 in 4 chance of getting this Punnett square, and because these are the same, if you wanted to know what the probability of getting a homozygous or heterozygous individual would be, it would be 50% in this generation, because you would take 1/4 plus 1/4, get 1/2. So you can't get this zygote formed with this and get this zygote formed in the same organism. So this is always going to be mutually exclusive. If you have this, if you get this A, then you cannot get this A, and if you get this A, you cannot get, or if you get this A, you can't get this A. If you get this A, you can't get this A. So it's mutually exclusive. You can only get this or this, but the probability of getting either of these would be 50%, because you could add them together. The rule of multiplication then is looking at events that aren't mutually exclusive. So that would be looking at what are the probabilities of getting a big R and a big Y, for example. The probability of getting a big Y and a little r. So in that case, you could actually do this problem in 2 Punnett squares, with one of the Punnett squares having one of the traits on them and the other Punnett square having the other trait. In this case, I represent it by A's and B's. So say, for example, you want to know what the probability of getting only dominant traits are. So you have this A trait and you have this B trait. If you want to know what the probability of getting dominant A, and dominant B, you would multiply these together. So you would have 1/4 of a possibility over here, 1 out of 1, 2, 3, 4. And you'd have 1/4 of a possibility over here, so that's 1 out of 1, 2, 3, 4. 1/4 times 1/4 is 1/16. If we go back to here, we can see that we have 16 blocks here. Only one of them has all of the capital letters in there. So this is how the rule of multiplication works. All right, I hope that was informative, and I will see you guys later.